Hi guys, so here's our second video about non-Mendelian inheritance. We've got codominance. Codominance is when the two alleles are both equally bossy, neither one covers up the other, and neither one goes into the background. So they both show up side by side, both visible in the phenotype. So in codominance, two alleles are both present in the phenotype, but they do not mix. They are not going to mix together to create an intermediate color. They're going to both be there side by side. So the heterozygous phenotype is going to be both phenotypes clearly visible. You'll see red and white. So in a cow, let's say we're going to go to the cow in the next picture. Here we go. Here's our shorthorn cattle. He is codominant with red and white hairs. So instead of mixing together and creating a lighter brown color, the homozygous red appears next to the homozygous white, and they are both dominant. They both show up. If you have a dog with spots or a cat with stripes, those are codominant alleles that work in this way, that they both show up side by side. Here's a horse that has less defined stripes, but as you can see, there are red hairs, there are white hairs, they are all intermingled on this horse. There are no pink hairs here. There are no lighter brown hairs that have been created by incomplete dominance. Both the red and the white have shown up. This is what it often looks like in flowers. If you see a striped flower or a flower with spots, these are codominant. The red and the white appear side by side. They are both dominant. They do not mix in codominance. In sickle cell anemia, this is a serious condition in which blood cells form a different shape and they cannot carry very much oxygen. It can be unhealthy. So these are codominant alleles caused by an abnormal hemoglobin. So normal hemoglobin has the RR genotype. The sickle-shaped blood cells are SS. Both are dominant. So people who are heterozygous carry the disease and they have a mixture of normal and sickle cell. And this is what the Punnett square looks like. You have normal brought down into these two squares and sickle cell brought down into these two squares. If this parent has two alleles for sickle cell, in the intermediate, the heterozygous genotype, you have both sickle cells and normal cells. And in the homozygous sickle cell, you only have this type of blood cells. So this is how the Punnett square works for a codominant trait. So let's stop and think. If there are two alleles for a hair color trait, red and blue, and it's incomplete dominance where they mix, what are we going to get? Red and blue mixing together in incomplete dominance would make purple. Red hair and blue hair combined in codominance, same question, different conditions. If it's codominance, you would get red and blue patches next to each other because neither of those covers up the other and they do not mix. All right, we're going to skip to page 25. Which one is codominant? Codominant meaning they appear next to each other. This one's codominant, both are equally present. This one is incomplete dominance where the red and the white combine to make pink. That is incomplete dominance with mixing. You can see more about this in the Amoeba Sisters video.